After Mendel did all of his studies looking at the 7 different traits of the pea plants independently, he then took it one step further and started to look at 2 traits at the same time. So these were dihybrid crosses, so 2 traits, and he set them up the same way. So taking pea plants that were round and yellow, homozygous, and then green, wrinkled, homozygous, produced an F1 generation, which is what we can see right here. That would be heterozygous for both traits, so that's why it's dihybrid. And then he looked at the F2 generation. So notice if we're looking at these plants right here, big R, big Y, little y, that's their genotype. The phenotype of each of these pea plants would be round, seed shape, and yellow pea. So round and yellow is their phenotype. So let's look at the results that Mendel found. All right, so up here, just showing here's our pea generation. So homozygous, round yellow, homozygous, wrinkled green. When those were crossed, they produced heterozygous, round and yellow plants. So when he crossed these, what he found is he didn't just get round and yellow and wrinkled and green. He got a lot of round yellow plants, 9 of them in fact. So 9 out of the 16 possibilities were round and yellow. He also got some that were round and green. He got 3 round green, 3/16 were wrinkled and yellow, and 1/16 were wrinkled and green. So you can see that based on the pictures, but also if we look at the genotypes. So for example, if we look at this pea right here, okay, it has the big R and a little r, so that tells you it's round. It has the dominant allele for shape, so it has round, but then it has 2 little y's, so that would make it green, so that's round and green. Now what he found for the phenotypic ratio, and he did lots of different combinations, always looking at these dihybrid traits, is that 9/16, 3/16, 3/16, and 1/16, so that was a 9:3 to 3:1 ratio. So if you ever see that 9:3 to 3:1, and things are in sixteenths, because if you add all of those up, it adds up to 16, that indicates you're looking at 2 different traits. Okay? This led Mendel to his second really important law. We talked about the law of segregation. Now we also have the law of independent assortment. And this is something we talked about earlier with meiosis. And this is the idea that the alleles separate or segregate independently from one another when they're on different chromosomes. So the chromosome pairs, remember we talked about how they could line up either way on the equatorial plate during metaphase 1, and how they line up determines which alleles end up in each gamete. And every allele combination, or every chromosome combination, is equally likely to occur. And this is when we used that formula 2 to the n. Remember, so in humans, it'd be 2 to the 23rd, because our haploid number of chromosomes is 23, so 2 to the 23rd, which is over 8 million different combinations of how the chromosomes can line up. So that's the law of independent assortment. What we're going to look at now is how to actually set up a dihybrid punnett square. This is something that you probably won't have to do on MCAS, but you may have to do it if you take AP Bio or genetics in the future. So let's try it out. So let's say you have to try to figure out the possible combinations in a dihybrid cross between 2 freckled tongue rollers who are both heterozygous for each trait. Okay. Now, a couple of rules when you're writing genotypes and dealing with more than one trait. First of all, you always want to keep the alleles for the same trait together and in the same order. So in this case, we would always write the F's first, then the T's. So you don't put FT, FT. Okay? You also list the dominant alleles first, but you still keep letters together. So what that means is if the genotype was big F, big T, I would write like that. If it was little f, big T, it would be like that. So you always keep the F's together and the T's together, so because those are the alleles for the same gene, so those want to be next to each other. And then it's just helpful to list the dominant alleles first, if they exist, because that makes it easier to figure out phenotypes. To figure out how to set up the punnett square, you need to know what the possible gametes are. And because each gamete is going to receive one of the chromosomes from this pair, so one F allele, right? One allele for freckles or no freckles, and one chromosome from this homologous chromosome pair, so one of the alleles for the tongue rolling, you need to look at the different combinations. I always use something from algebra, the FOIL method, first, outer, inner, last. Basically what that means is I'm going to take the first combination, so big F, big T, then I'll take the outer pair, right? Big F, little t, then the inner, little f, big T, and then finally the last, little f, little t. Notice that there are now 4 different possible combinations, right? 1, 2, 3, 4. They are all unique. So if you think back to when we were doing meiosis with pipe cleaners, and you had the pink and the blue pipe cleaners, and when you separated them out, you could get different combinations depending on how you lined them up. That's what we're looking at here. Now that we figured out the gamete possibilities, we're going to put those in the outside of our punnett square, because that's what goes on the outside, right? All of those different possible gametes, which is a little bit simpler when you have only 2 possibilities, but when you're looking at 2 different traits, now all of a sudden we have 4 possibilities. Since both parents have the same genotype, the combinations are the same on both the top and the bottom. So now we figured out our gamete possibilities. So what we're figuring out in the middle are all of the possible combinations that could occur. Each one is just as likely as the next. So I'm going to, go across and down, just like we do with our punnett squares. And I'm going to write my F's together. So big F, big T, big T. This one would be big F, big T, little t. A big F, a little f, big T, big F, little f, big T, little t. Okay. So I'm going to keep filling this out, and I would like you to do the same. All right, so now that you have your whole punnett square filled out, what we need to do is now interpret the results of this. So what does it all mean? So I'm going to color code everything. So I'm going to shade in with this kind of reddish color all of the individuals who are dominant for both traits. So they have at least one dominant allele for freckles and at least one dominant allele for tongue rolling ability. So I can see if I go along, all of these do, this one does, this one does not, but this one does. And we go through all of these. So I can see that 9 of the individuals are dominant for both traits. Now we'll do the same thing. I'll use a different color to code all of the individuals who would have freckles but not be able to roll their tongue. So have at least one big F but 2 little T's. So we can see this one right here, here, and here. So 3 out of the 16. And let's do the same thing for no freckles, so 2 little F's, but at least 1 dominant allele for tongue rolling. So we have 1, 2, 3, again. And then this last one, which has all recessive alleles, so no freckles, no tongue rolling, I'm going to leave that one just white. But now we can use this to answer the questions on the next slide. All right, so how many were freckled tongue rollers? So that would be dominant, at least 1 dominant F and 1 dominant T. Okay, that was 9 out of 16. Okay, how many were freckled? So at least 1 dominant allele, but non-tongue rollers? That was 3. How many were freckleless? Not a word you hear all that often, but able to roll their tongue? That was also 3. And then the freckleless non-tongue rollers, there's only 1 of those, right? And this is out of 16, and that's, again, our 9 to 3 to 3 to 1 ratio.